morning to all of you, and I am back again with another lecture which is based on staining methods. We have seen negative staining, we have seen monochrome staining, we have seen acid fast staining in my last lecture, and today's lecture will concentrate on an important point, that is gram staining. Now before we go to gram staining, let us understand what are the measurements, why organisms that we see are so small and how they are being measured. Based on that, the microorganisms are very small. In my previous lecture also I told you that microorganisms are those which cannot be seen with the help of a naked eyes, and therefore we need some kind of instrument to see those microorganisms, that is nothing but a microscope. So they are very small. They use the metric system to understand or to see the microorganisms. Metric or meter m standard unit, micrometer mu m, this is called as mu m, is equal to 1 into 10 raised to minus 6 meters. Bear in mind, nanometer is again a scale which is denoted by nm, and 1 into 10 raised to minus 9 meters. This will be the thing. Angstrom will be 1 into 10 raised to minus 10 meters. So this will be the measurement part. Now, bacteria have nearly the same refractive index as water. Therefore, when they are observed under the microscope, they are opaque or nearly invisible to the naked eyes. So bacteria are so small that they cannot be directly seen with the help of a naked eye. So what one can do is that they have the refractive index which is similar to water. So if you put a drop of water and if you observe under the microscope, you will never see any sort of microorganisms in that water directly. So in order to see those microorganisms, a method called as staining has been developed, and this staining method will be helpful for observing the microorganisms under the microscope. So different types of staining methods are used to make cells and their internal structures more visible and under the light microscope. Now what is the stain? So some basic part again this is, we will try to revise it and then we will come to the important part. Which are a chromophore groups which is conjugated double bond and give the dye its color, which can bind to cells by ionic, covalent or hydrophobic bonding. For example, a positively charged dye binds to negatively charged structures. We have already seen this part in case of monochrome staining as well, that the cell surface will certainly have a charge, and based on that charge, the type of stain that we are using has to be decided. So once that decision is being made in a final way, then and only then one can that stain will easily penetrate inside and can work very easily. So an isotope has been divided into two categories: basic dyes and acidic dyes. We have already seen this part, so I won't be going in detail of the same. Now we come in one of the most important part. Differential stains use two or more stains and allow the cells to be categorized into various groups or types. Okay. Then this technique allows the observation of cell morphology or shape, but differential staining usually provides more information about the characteristics of the cell, that is the thickness of the cell wall, so and so. So differential stain, it uses more than two stains and it helps in what? A different differentiation between the two types of bacteria that are being cell. Now this technique was developed by a great scientist called as Graham, and he has named himself after this particular kind of technique. One is called as, his name is Hans Christian Graham, and differentiate between gram positive purple and gram negative pink stains and is used to identify certain pathogens. So for this purpose one has to undergo with this particular type of method which is developed by Dr. Hans Christian Graham. Now this is most important. If you see the structure of the two types of bacteria that we have, one is called as gram positive bacteria and gram negative bacteria. Okay. Now you see the peptidoglycan layer over here. Just mark it, and the lipopolysaccharide present into the gram negative bacteria. See this. Differentiation between the structure of the cell wall of gram positive and negative. We will come to this slide once again once we understand this fully. Now, what is the principle of this technique? Gram staining is used to determine gram status to classify bacteria broadly. It is based on composition of the cell wall. So what is the composition of the cell wall? Based on that, the classification of these two stains has been done. Gram staining uses crystal violet to stain cell walls, iodine as a moderant, and fuchsin and safranin combusted to mark all the bacteria. So there are 4 different types of stains that are being used in this technique. One is called as crystal violet, second iodine as a moderant, before safranin it is the decolorizer, that is alcohol, and ultimately safranin, which is being helpful for it as a counter stain. So gram staining is important in medicine. What is the application of gram staining? In presence or absence of cell wall will change the bacterium susceptibility to some antibiotics. Gram positive bacteria stain dark blue or violet. Their cell wall is typically rich with peptidoglycan and lacks the secondary membranes and lipoperoxylated layer found in gram negative bacteria. So we come to this slide back again. See this. The peptidoglycan layer in terms of gram positive bacteria. See how the density of the layer is. कितना ज़्यादा बड़ा layer है वो, that you can easily identify over here. And here if you try to understand, see this is for the gram negative bacteria. So gram positive bacteria have a higher range of peptidoglycan with teichoic acid polymers which is present inside this particular layer or membrane of the cells. Then now these are different stains that are being utilized. What we have crystal violet staining solution. That is called as the primary stain. Primary यानी पहली बार डालने वाला जो stain आता है, that is primary stain. Gram sardine solution called as moderant. 70% alcohol solution called as decolorizer, and safranin staining solution as counter stain. So ध्यान में रखने वाली चीज है that there are 4 stains. Actually there are 3 stains. One is called as the primary stain. This is moderant, which is an iodine solution. Decolorizer 70% alcohol, and counter stain, which is safranin, that has been utilized. If you visit the laboratory, you can easily see all the bottles which are present inside the laboratory during the practical. Now the procedure. See this. 1, 2, 3, 4 stain है. For that purpose, what we are going to see? Crystal violet. First, make a smear, and onto that smear, just flood the slide with crystal violet. Wash it with the help of a water, so that the excess amount of water which is present over there will be removed out. Then flood the slide with the help of grams iodine. Again wash it a little bit amount of water, so that the excess amount of iodine which is present will be removed out. Now use the decolorizer. Very small amount of decolorizer is being utilized. If you go along with some excess amount of decolorizer, then all the stains may be washed down. So one has to be take care that the decolorizer should be in a very small quantity. With a drop by drop you can decolorize the cell. Then again it has to be washed with water. Very gently you have to wash. Counter stain with safranin. Secondary stain or the counter stain which is present safranin, you have to flood the slide, and again wash after a couple of minutes or couple of time, and then you will be able to observe, air dry it, and you will be observe the same. See this. Crystal violet. Once we put on crystal violet, all cells which are present will be colored as violet in color. Then you add iodine as a moderant, which fix that particular color into gram positive bacteria. Then you go along with alcohol wash, that is decolorizer. Now once you put alcohol into it, the alcohol will act on the lipopolysaccharides or the peptidoglycan layer which is present over there. But if you come to this slide once again, gram positive में कैसा था? The layer is very hard or very big. So when you are utilizing the decolorizer, this membrane will not be ruptured for a long period of time, and therefore once the stain is inside the cell, it won't go back. Therefore, instead of this, if this cell wall, if you observe here, this cell wall have a layer which is the very low layer. What we can say, the complexity of the layer is not there. So there and therefore, once the decolorized layer, the membranes here are been ruptured, or they allow the crystal violet to be washed down. So this figure, if you consider, here you can see that some of the cells, there, because of the decolorizer, the stain has always been removed, while the other stain has already retried. So ये कौन सी cells है? These are gram-positive cells. Why? Because the layer of peptidoglycan is very high as compared to gram-negative, and therefore when you put down the counter stain into it, after counter staining you will see that the layer or the cells are being colored with different color, which is of the counter stain. यानी इसके बाद में क्या होता है कि once you put in the decolorizer, the flaps or the pores are open, therefore the decolorizer will help the stain to move out. And again when you are counter staining, the red color stain, that is saffron, which will come inside, and the cells will appear to be red in color instead of blue in color. Now these are those cells which have a higher range of peptidoglycan which is present, which are gram positive. Gram positive क्यों बोला है? Because it has been counter stained, it has been stained with the help of the main stain, that is the primary stain which has stained it. See this, इसका color कभी change नहीं हुआ है. इसका कलर अभी कभी तक हम लोगों ने चेंज नहीं देखा है. Instead of that, if you see these bacteria, primary stain वहाँ पर है. After moderate and after decolorization, the color is not here. And when you are counter staining with the help of saffron, the color appears to be in this fashion. And therefore, one can easily understand the basic of this technique. See this is actual pictures that we have taken. This is gram-positive bacteria. If you see the color of these bacteria, this is very similar. These are gram-positive and gram-negative bacteria. ये देखो, जो रेड कलर में दिख रहे हैं आपको, these are gram-negative bacteria. These are gram-positive bacteria. The violet color bacteria should be gram-positive bacteria, while the other bacteria should be gram-negative. See, negative cocci, gram-negative rods. ये लेयर का डिफरेंशिएशन देखिए यहाँ पर कैसा दिया हुआ है. See the layer in gram positive bacteria. ये लेयर ज्यादा होने के कारण once you put out the alcohol, they will not be able to dissolve the entire layer, and therefore the counter stain जो counter stain जो बाहर में है वो अंदर नहीं आ सकता है. और अंदर का जो क्रिस्टल वाले का स्टेन है, that even that won't be allowed to go outside. लेकिन जब ग्राम नेगेटिव में आता है, ग्राम नेगेटिव का कैसा है लेयर देखो, छोटा सा लेयर है, so it can be easily dissolved with the help of a decolorizer, and the counter stain would come inside, and this original stain will go outside, and therefore it appears to be red in color. Gram positive bacteria are those that are stained with dark violet by gram staining. This is the contrast to gram negative bacteria which cannot retain the crystal violet stain instead of taking the counter stain. जो मैंने अभी बोला वही हम लोग यहाँ पर देखते हैं. And appearing red or pink. Gram positive organisms are able to retain the crystal violet stain because of the high amount of peptidoglycan in the cell wall. उनकी cell wall की जो रचना है उसके basis के ऊपर the staining method has been developed. इतना बढ़िया से साइंटिफिक इन्वेंशन है कि एक ही बैक्टीरिया या बैक्टीरिया का मिक्स्ड कल्चर अगर है तो उसमें भी हम लोगों को डिफरेंशिएट करते आएगा whether it is gram positive or gram negative based on these aspects. See this, in terms of gram positive bacteria, characterized by having a part of the cell wall structure peptidoglycan as well as the polysaccharide and teichoic acid, that is peptidoglycan is sometimes called as murines or heteropolymers of glycans. ये ग्राम पॉजिटिव बैक्टीरिया का है. ग्राम पॉजिटिव बैक्टीरिया में कैसे लेयर्स है पेप्टिडोग्लाइकन हायर अमाउंट में एंड टेकोइक एसिड दैट इज बीइंग प्रेजेंट ओवर देयर एज वेल. नाउ ग्राम नेगेटिव बैक्टीरिया retain crystal violet dyes in gram staining protocols. In gram staining test, a counter stain saffron is added after the crystal violet and gram negative bacteria will appear red or pink. The test itself is useful in classifying two distinct types of bacteria based on structural differentiation of the cell wall. ये देखो, ये ग्राम पॉजिटिव और ग्राम नेगेटिव में इस प्रकार से. See this is in terms of gram negative bacteria, and see the layers of the LPS or the lipopolysaccharide which has been present over here. Some examples. ये बहुत important slide है. Importance of the gram positive bacteria. You can see the cocci and the rods. Staphylococci, Streptococci, Enterococcus, Peptidos, Streptococcus, Corynebacterium, Listeria, Lactobacillus, Nocardia, Mycobacterium, Bacillus, Actinomycete, and Clostridium. These all bacteria are said to be the gram positive bacteria. While when we come in this side, the gram negative bacteria may be Pseudomonas, Salmonella, Shigella, Klebsiella, Proteus, Escherichia, Yersinia, Protella, Haemophilus, Brucella, Pasturella, Vibrio, Bacteroides, Fusariums, and Protoloans, and Leptospira and Bordetella are also been classified in that way as well. So these are certain examples of positive and gram-negative bacteria. Correct? So these are certain references. You can use these references for your further studies or for your further readings. Correct? So that's all for today. We'll stop here and we'll come with the next part of the syllabus, that is the staining. Staining, we have finished this part. We'll come to the media or bacteriological media which is present and how to culture organism in my next lectures. Sure. Thank you so much.